Okay, we're live and we're recording. Welcome everyone to today's fourth episode of The Future of Foraging. Our topic today is food for thought, how internal states shape foraging behavior. And we have two really fantastic speakers for you today, Dr. Audrey Dussutour, as well as Dr. Ron Gong. And we also have a guest panelist, like last time, which is Charlotte Rocher, a PhD student at the Champollion Research. And just to start off, I will briefly introduce both of the speakers before we start with the first talk by Audrey Dussutour. So it's my pleasure to introduce our first speaker, Dr. Audrey Dussutour. She is the CNRS Research Director at the Paul Sabatier University in Toulouse, France. In 2004, she received her PhD degree in animal behavior working in the laboratories of Vincent Focassier at Paul Sabatier University, as well as Jean-Louis Stenneberg at the University of Brussels. She then conducted postdoctoral research at Concordia University in Canada with Emma Desplant, and at the University of Sydney in Australia with Steve Simpson. Since 2009, she has a permanent position at the Research Center on Animal Cognition in Toulouse. Her current research work focuses on protocognition in slime molds, as well as nutritional state-dependent collective decision-making in ants, which she will also present in today's seminar. Our second speaker today is Dr. Ron Gong. Much of her work in her career thus far has been focused on the understanding of the neurobiological mechanisms underlying various psychiatric disorders. Dr. Gong began her studies in Wuhan University in China with a BA in biotechnology. She earned her PhD from the National Institute of Biological Science at the Tsinghua University in Beijing. She then worked with Minmin Lu to study the role of membrane receptor guanylyl cyclase C in attention deficit hyperactivity disorder, and also investigated causal role of hindbrain norepinephrine neurons in anxiety. From there, she moved to Genelia to Scott Stearnson's lab, where she has focused on internally motivated behaviors for consumption and the role of specific populations of neurons in the paralocus coeruleus in such behaviors, and that's what she's going to talk about today. So with that, I would like to give Audrey the stage, and I will now close the videos of the others. Thank you.
Thank you very much, Denis, for this kind introduction and for this invitation. So I'm going to try to share my screen. Okay. Everything is fine? I guess yes. So today I'm going to talk about how ant colonies solve complex nutritional challenges. So first of all, nutrition is complex because achieving nutritional homeostasis to maximize fitness involves a complex interplay between multiple and changing nutrient needs and variable foods. But nutrition is even more complex when sociality is mixed into the problem. So sociality complicates nutritional regulatory strategies, and it's what I'm going to try to demonstrate today. So the question we have been trying to ask for the last 10 years is, how do ants adjust their foraging and feeding strategies to nutritional constraints? So the first problem in ants is that the food entering the nest is collected by a small number of workers and is shared among all members of the colony. So only 10% of the ants are going outside, collecting food and sharing with the others, sometimes via trophallaxis. The second problem is that ants within the nest have different nutritional needs. Workers need carbohydrates as a source of energy, while larvae needs protein to grow. So foragers need to relate to the nutritional demands of the nest as a whole and themselves as individuals. So how do ants colony balance nutrient intake for the whole colony when they go out and forage? So to answer this question, I use a geometric framework, which is a state space modeling approach to deal with nutrition complexity. It was first applied to nutritional ecology of locusts in 1993 by Steve Simpson and David Robenheimer, and since then it's been applied from slimes to human. So the first thing you need to do to study nutrition following the geometric framework is to design an artificial diet. So this is what we did with Steve, to design a diet where we could manipulate all the nutrients individually. And the first question we asked was, what are the amount and balance of nutrients that maximize the colony fitness? We wanted to find what was the optimal diet. And to do that, we confine ants to one of multiple diets differing in the ratio of protein to carbohydrates, and we measure the longevity. Longevity is a proxy of fitness, but we will see later that there's other proxy of fitness. So here, this is what we obtained for a species called Lasus niger, which is a native species in France. So here you have the protein collection, here you have carbohydrate collection, and here it's what we call a performance landscape, showing the lifespan of ant on 16 different diets for 130 colonies, and so it was more than 26,000 ants that we followed. And we found out that the optimal diet was a 1-4 diet, so 4 times more carbs than protein. So here you can see that lifespan is optimized here. We ran the same experiment with Sarah Arganda on an invasive species called Inepithema humile, or the Argentine ants. And here we played with 12 different diets, 100 colonies, and 20,000 ants. And we show also there was an optimum here, and the optimal diet was a 1-5 diet, so 5 times more carbs than proteins. What you can see when you compare this performance landscape straight away is that the optimum for the two species is not as wide as for this one. For instance, for Lasus niger, the optimum is quite restricted in the nutrient space, while it's quite wide for Inepithema humile. So recently, in a review published in Trends in Ecology and Evolution with Jonathan Cheek, we argue that, in fact, the width of this fundamental nutritional niche will play a role in the invasive potential of species. So let me explain. So for instance, if you place these two species in environment A, offering these five food sources differing in their protein to carbohydrate ratio, the species can reach easily its optimum here. And it would be the same for Inepithema humile. If now we place this species in environment B, so here offering only these 4 food sources, you can see that Lasus niger cannot reach its optimum anymore, while Inepithema can still reach its optimum. So we discussed this effect on 100 different invasive species from insect to mammals in this review. So as I say, longevity is one proxy for fitness, but there's other proxy, and one that is important is reproduction success. So here it's a study conducted by Kwang Poom Lee in Steve Simpson lab on Drosophila, and it shows that lifespan is maximized on a 1-16 diet in flies, but reproduction success is maximized on a 1-4 diet. So animals have to do a compromise between maximizing lifespan and maximizing reproduction. So we run the same experiment with ants, and we picked a clonal ant called Platytheria punctata. So it's a clonal ant, so each ant can give birth to another ant, would be completely identical genetically. So here you have the number of eggs produced, and here you have the longevity, and you can see that in fact in ants, it's very different from flies because longevity and reproduction success are maximized on exactly the same diet, a 1-1 diet. So this work was done in collaboration with Abel Bernardo and Mathilde Vidal in Regensburg University. So the question we can ask also when it comes to ants is how the balance of nutrient influence division of labor. As I said earlier, foragers are foraging for food, the queen is producing the eggs, and the nurses, for instance, take care of the larvae. So what we did was to manipulate ants' nutritional state and see if it affects division of labor. So we first conducted this experiment on Platytheria punctata, the clonal ants. So ants were placed individually and confined to one diet, here a 1-1 diet, so high protein diet, or on the 140 diet, so it's a high carb diet. So these ants were confined individually for 2 weeks, and after we built pairs, and these pairs were stayed together for 6 weeks with a completely balanced diet. And we looked at who take in charge the reproduction. So to do that, we dissected the ovaries of the ants, and we show that the ants that were fed on a high protein diet had a higher probability to become the reproductive, while the ones that were fed on a 140 diets had a higher probability to become, in fact, a forager. Only 29% became the reproductive individual in the pair. We ran the same experiment, this time focusing more on the worker cast with Steve Simpson. And what we did here is to manipulate both protein, carbs, and fats. So we created a high-fat diet and a low-fat diet, and we confined our ants for 4 weeks. And after we mixed them together, so they were the same age, the only thing that we manipulated was the nutritional status. And we looked at who became forager. And in fact, the fat ants, didn't stay inside the nest, while the ants that were fed on a low-fat diet became forager. So the next question we asked was what to eat when the optimal diet can't be reached. So in the next experiment I'm going to show you, we forced ants to ingest diets that are in balance to confront them with a conflict between meeting their requirement for protein and carbohydrates. So here, ants, they cannot reach their optimal diet. They have no choice to compromise. So we tested this experiment on different species, Lasius niger, argentinense, Linepithema humile, Rhytidoponera sp, and Odontomachus. So what we show that if ants are confined on different diets, varying in their protein to carbohydrate ratio, what ants do first is to satisfy their requirement for carbohydrates. But to do that, like you can see here, they would suffer an excess of protein or a deficit of protein. Indeed, to reach this intake target in terms of carbohydrates, ants have to eat a large amount of this diet. But by following this strategy, of course, this excess of P comes at a cost. And we measure this cost of eating an excess of protein. So we measure this cost in terms of longevity. So here it's on the species Rhytidoponera metallica. So here you can see in red the high protein diets, and in blue the high carbs diets. So you can see that ants die very quickly on a high protein diet, and they almost didn't die on a high carb diet. We found exactly the same pattern in Lasius niger. So here you can see the high carb diet, ants live for very long, almost 400 days. While on a high protein diet, they didn't live for very long. We found exactly the same pattern also in Linepithema humile, the argentinense, and they didn't live long on a high protein diet, but they live longer on a high carb diet. So with Sarah Arganda, we were wondering if this toxicity of protein came from the digestion of the protein itself, or in fact, the amino acid after digestion. So what we did was to translate all these diets in amino acids diets. So here we use whole protein, and here we use amino acid diets. And so what we could see is that, in fact, the effect was still there, and it was stronger. Ants died quicker when the food were made with amino acids. So then after, with Sarah, we fed our ants a lot of different diets where each of the amino acid was in excess. And we show that methionine, serine, threonine, and phenylalanine were the worst amino acid when it came to survival. So then, knowing that ants can make compromise, we wanted to see how they solve nutritional challenges. So the first challenges was about concentration, protein and carbohydrate concentration. So what we did was to offer ants diet with different P plus C concentration and looked if they were able to achieve the same nutrient intake. So here, so this is an example in Lasius niger. So what we did was to use a very concentrated diet. So it's a high carb diet. And this diet was then diluted 5 times, and we looked at the amount of food collected by the ants. And we show that the foragers were able to collect 5 times more food when the food was diluted 5 times. So they were able to solve this nutritional challenge and cope with nutrient dilution. The next nutritional challenge we faced them with was colony composition. As I said earlier, ants and larvae have different nutritional needs, and we wanted to see if it could affect how ants solve nutritional challenges. So we manipulated the ant colony composition and offered them a choice between two complementary diets. So ants had to feed from a 2-1 diet, so high protein diet or high carb diet. And so they were first alone by themselves, so it was only workers. And we show that ants prefer to feed on mostly on 1-3 diet, so they had a high carb intake target. While if we added larvae in the colonies, we could see that ants were feeding mostly on the 2-1 diet. So they changed, in fact, the optimal diet as a function of the presence of larvae inside the nest. So they were able to change their foraging strategy according to colony composition. So now I'm going to spend more time on this last study where we manipulate the ants' nutritional state and offer them a choice between two complementary diets. So here, what is the best food source? I wanted to show that it depends on the nutritional state of the ants. So what we did was to confine colonies to a nutritionally defined diet from which we removed one or more nutrients for 5 to 10 days. And then ants were allowed to select between two food solutions offering the missing nutrient or another non-missing nutrient in a 1-hour choice assay. So it was a very quick experiment. So ants needed to compensate for the deficiency incurred only in 1 hour. So here it's how the experiment looked like. So ants are offered two food sources, and we looked at their feeding behavior. And you can see here ants exchanging food directly on the bridge via trophallaxy. So first we measured at the collective level. So we run 22 different choice experiments, and we run almost 500 assays with more than 180 colonies. All ants were counted, so all the ants that went on each in the direction of each food source were counted. And then at the individual level, we followed more than almost 10,000 ants, and we looked at if the ants were feeding or not once they reached the food source. So the first experiment, we created deficiency in carbohydrates and essential amino acids. So here you have the deficiency that ants were faced with, and here it's a proportion of ants on each food source. So these are the results at the collective level. So here you can see that when ants do not suffer any deficiency and they were offered a carbohydrate food source or an essential amino acid food source, ants would prefer to collect food from the carbohydrate food source. If now we created a deficiency in carbohydrates, you can see that the preference is still there and it's even stronger. Ants almost went only to the carbohydrate food source. When we created a deficiency in essential amino acids, we can see that here there's almost no differences and ants start to collect essential amino acids. And when ants were deficient in both carbs and essential amino acids, you can see that the preference for carbohydrates also decreased. So if we now look at what's happening at the individual level here, so here the numbers are the probability of feeding on the food source at an individual level. So, for no deficiency, only 50% of the ants fed ants at the food source, and only 35% of the ants for the essential amino acid. When we created a carbohydrate deficiency, this probability of feeding increased up to 92% of the ants feeding, but stay very low for the essential amino acids. When we created an essential amino acid deficiency, you can see that both probabilities are equal. And when we created both an essential amino acid and carbs deficiency, you can see that both probabilities are high, and they were higher for the carbohydrate food source. Then we run the same experiment, but this time we didn't play with carbs, but we played with sterol and essential amino acids. So ants were either deficient in sterol or in essential amino acids. And what you can see is that when ants are deficient in sterol, they prefer to collect food from the sterol food source, while it's the opposite when they were deficient in essential amino acid. So this was at the collective level. And at the individual level, we saw exactly the same thing. The probability of feeding was higher on sterol when ants were deficient in sterol, but was higher on essential amino acid when they were deficient in essential amino acids. Because in the field, food are seldom just a pure carbohydrate food source, and they usually offer a mix of different nutrients. In this third experiment, what we did was to create a deficiency in both carbs and essential amino acid, and offer either a pure food source of carbs, so one that the ants really like, or a mixture of essential amino acid and carbs together. And we manipulated the ratio of essential amino acid to carbohydrates. What you can see is that when the ratio of essential amino acid is low, so it's meaning that there's more carbs than essential amino acid, ants prefer the mixed food than the pure source of carbohydrates in these two experiments. But when the ratio is biased toward essential amino acids, the carbohydrate preference comes back. So if you look at the individual level, we obtain exactly the same. When ants were deficient in carbs and essential amino acids, they preferred to feed on the mixed food source when the ratio was low. But as soon as the ratio becomes higher in essential amino acids, ants prefer to go and feed on the carb food source. Since then, we had been playing only with essential amino acid, and in this experiment, we wanted to see if ants could make a difference between non-essential amino acids and essential amino acids. So in this experiment, ants were deficient in carbs and all the amino acids, and we offer them a mixture, offering either non-essential amino acids and carbs or essential amino acids and carbs. And we varied the ratio of amino acids to carbohydrates. What we could see is that at the collective level, all the time the ants would prefer to forage on the food source offering essential amino acid over non-essential amino acid. And we found exactly the same results at the individual level also. The probability of feeding was always higher on the food source offering essential amino acids than non-essential amino acids. So you can see here the numbers always above here, and here they were very low. Knowing that they could differentiate non-essential from essential, we went further, and we wanted to know if they could differentiate amino acid between each other. So first, we created a deficiency. So ants have everything in their diets except 5 amino acids. So we picked them randomly. So the first mix was methionine, histidine, valine, arginine, and threonine. And the second one was tryptophan, isoleucine, leucine, phenylalanine, and lysine. So when the first mix of amino acid was missing, they went directly to it, like here. And when the second mix of amino acid was missing, they went straight to it. So they were able to differentiate this subset of essential amino acid at the collective level. And if we look at the probability of feeding at the individual level, we found exactly the same. When the first mix was missing, the probability of feeding was higher here. But when the second one was missing, it was here where the probability of feeding was higher. So the individual level always follows the collective level. Then we were trying to make things even more complex, and this time we created deficiency in only 2 essential amino acids. So we tested arginine, threonine, and histidine, valine. So here, results at the collective level are not significant, but we can see a tendency. Ants prefer to go on the essential amino acids that are missing in both cases. At the individual level, it's significant. Ants prefer to feed on the essential amino acids that are missing. So here, the probability of feeding is higher than here, and it's the opposite when it's the second subset of amino acid missing. So we ran this experiment twice with a new dose of essential amino acid, and this time we created a deficiency in methionine and threonine, or in tryptophan and phenylalanine. And here at the collective level, the results were significant, and ants still prefer to go to the food source offering the missing essential amino acid in both cases. And at the individual level, again, we found exactly the same results. Ants prefer to feed on the essential amino acid missing. So here the probability of feeding is higher than here, and it's the opposite when we create the opposite deficiency. And in the last experiment, because ants was so good, we decided to create a deficiency in a single essential amino acid. So here you have to imagine that the ants have a food source completely defined. Everything is there except one essential amino acid. So we either removed leucine or methionine. So when we removed leucine, ants were not showing any preference for any of the food source. But when we removed methionine, ants prefer the food offering methionine. And at the individual level, both probability were different. When leucine was missing, ants prefer to feed on a food source offering leucine, while when methionine was missing, ants had a higher probability of feeding on a food source offering methionine. So these results are quite interesting because This is just a 1-hour assay, and it seems that ants were able to cope with this deficiency quite easily. To understand what was happening, we built a model. So based on our data collected as the collective behavior, meaning the flow reaching each food source, based on the individual level, so the probability of feeding, and after we run simulation. So first, the first rule in the model that you have to know is about trail laying behavior. So in ants, when they're going out in the field and collect food source, they lay chemical trail on the ground. And these chemical trail are very attractive for the congeners or reinforce this trail. So you have seen, I'm sure, these trails in the environment and sometimes in your kitchen. So I'm going to go through the model very quickly. So the first rule was ants deposit a quantity Q0 of pheromone. So it was called passive marking, because in Argentine ants, wherever they go, ants are laying a trail. And the second rule was an ant that feeds add a quantity Q of pheromone, which is called active marking. It's true that when ants find a food source, they lay more more pheromones on the ground when they're more than when they are just exploring. So this gave us two differential equations showing the concentration on branch 1 and on branch 2. And so the question was, do ants modulate Q depending on food quality, or does only the probability of feeding P play a role? Indeed, because we could just have a very all or nothing rules, meaning that if you feed, you lay a trail, or depending on the quality of food source, you change the amount of pheromone you deposit. So our hypothesis was only P plays a role and Q is fixed, so that ants use an all or nothing rule. So it means that if they feed, they lay a trail. So we first needed for the model to calibrate Q and N, which is the steepness of the choice function. So what we did first was to compute the predicted flow on each branch for each treatment for each pair of Q and N. And we measure the error between predicted and observed flow on each branch for each treatment. Then we computed the total error for each pair Q and N. So big error are like big dots here. And we choose the pair with the minimal error and checked how the prediction aligned with observed flow. So here it was where the error was minimum, which is quite interesting because in previous model of Argentinians published in the 90s, they found exactly the same N equal 2. So we run the simulation, and I'm going to go very quickly through them. And so here are the experimental results we found, and in black, you see the outputs of the model. So here it's for experiment 1 with deficiency in essential amino acid, here deficiency in carbs, so it works pretty well. It works pretty well for deficiency in sterol and deficiency in essential amino acid against sterol too. It works also when we manipulated pure versus mixed food source. When we manipulated essential amino acid and non-essential amino acid, you can see that each time the model fits pretty well. It fits also well for the subset of essential amino acids that you can see here. With the duo of essential amino acid, the 2 duos, and the last experiment with the deficiency in 1 single amino acid. So, but to understand fully the story, we wanted to know who's making the decision when it comes to foraging. So in ants, usually extraneedle workers, the foragers, recruit intraneedle workers, the ones that are inside the nest, once they have found a food source. So usually extraneedle workers, the ones that are going out first, return to the nest and they hold information about the food. Whereas the intraneedle workers, which are in the nest, when they leave the nest, they hold information about the colony's nutritional states. So to prove that, what we did was to create nutritional conflicts within the colony. So in fact, we wanted to know what happens if everybody in the colony has not the same nutritional states. So what we did was to first differentiate extraneedle from intraneedle using fluorescent paint. So you can see here a lot of different ants painted with different colors. And so what we did was to have colonies of that we painted in different colors, and we created differential deficiency. So to do that, we had colony deficient in essential amino acid, colonies deficient in carbohydrates, and we swap the extraneedle workers, and so to create a conflict. So in the first experiment, extraneedle workers were carbohydrates. They had a deprivation in carbs, and intraneedle workers, the one inside the nest, were deprived of essential amino acids. So here I'm going to go through this graph. So here you have the time of the experiment, and here you have the proportion of ants going to each food source. So what you can see first is that ants are going first for the carb food source, and after it looks like they change their mind and they go for the essential amino acid food source. And here in black, it's the proportion of extra needle workers participating in the experiment. So what you can see is that when this proportion is quite high, they go for carbs. As soon as this proportion decreases, meaning that the intraneedle workers are coming out, they change their mind, and the intraneedle workers are following their own nutritional states. And if you look at the individual behavior, the probability of feeding, you can see that extraneedle have a higher probability to feed on carbs than on essential amino acid, while it's the opposite for intraneedle. So after we did the reverse, extraneedle workers were deprived in essential amino acids, and intraneedle workers were deprived in carbs. And you can see exactly the same effect. A first choice coming from the extraneedle workers, and after, when they don't participate anymore, the intraneedle workers are taking over and trying to satisfy their own nutritional states. And you can see that here you have the same probability. Extraneedle workers prefer feed on essential amino acid, while intraneedle prefer to feed on carbs. So this experiment shows that even when you create conflicts, the colony is able to answer. And so it means that the ants are following their own nutritional state when they go out and forage. And if they're not happy with the food source that is coming back to the nest, they will go out and change the way of it. So this is my last slide. I hope I convinced you that ants are a very good model and that they can solve complex nutritional challenges for the colony by making quite simple adjustment to the feeding and foraging behavior. As you can see, the rules are very simple. You only feed as a function of your nutritional state, and this would affect the whole colony as a whole. So I wanted to thank all the people who have been working on these projects with me. And Nico Xata was doing a postdoc, Sarah Arganda, Sepide Bazazi. Mathilde Vidal was doing a master and a PhD degrees. And of course, I want to thank my colleagues, Jonathan Cheek, Jacques Gautry, who built the model, Abel Bernadou, and of course, Steve Simpson. Thank you for your attention.
Thank you very much, Audrey. This was really an exciting talk. So we have a few questions already. So let me start with the first one. So Aditya Raju Gopalan asks, how does nutritional balance affect division of labor in animals that don't show this division at the level of individuals like ants do? Any animal that has to both forage for its own and a mate.
Oh, so you mean like a fly, for instance, when there's no division of labor, but he has to be like gregarious animals.
Maybe
where there's not for instance, in flies, they can do kind of a collective decision too. Like Mathieu Lioro shows that they can forage on the food source that maximize their lifespan as a collective too. But is not as integrated as in ants, where you have, in fact, in ants, what is particularly interesting is that reproduction is separated from foraging, in fact. So it's kind of the trade-off kind of disappearing in our clonal ants, but it would be very interesting to see in a colony more advanced, because our clonal ants are quite primitive, and it would be very interesting to see for instance, in a colony where the queen is very different from the workers, if they have the different, if this trade-off appears again, like in flies, for instance. I don't know if I answered the question.
Yeah, I really find it fascinating. How the internal state or nutritional state information is divided from the food sensing information in the two different worker types. It's really interesting. So let me see whether we have any further question in terms of clarifying things. Do the ants sense the amino acids by taste or by metabolites? How quickly does preference emerge during the assays? This is asked by
quick, actually, because if you look at the dynamic, it's done in after 10 minutes. So I have no idea, to be honest, about the physiology. This has to be like investigated by somebody who would be specialized in physiology. But from the behavior, what we can see, it can't be post-ingestive effect because it's too quick. So it has to be, I think, a sensing effect. So I don't know how they do it, but to be honest, I don't think it would be post-ingestive effect because I think it would take longer to have this. Because collective choice, in fact, the story is done after 10 minutes. After 10 minutes, you have the bifurcation and you have the collective choice. So I don't think it's a post-ingestive effect.
Okay. Let me see. So, apart from lifespan, did you also look at the effect of high protein diet on, oh, now it's moving, on the number of eggs produced in the colonies of the different species?
We did it only for the green-headed ants, and we show that the number of eggs was higher on for instance, it was on the 1-1 diet, but lifespan was better on a 1-2 diet. So it was not completely aligned like in the clonal ants. And I know that Jonathan Schick has been doing it on Mosmania, also the Pharaoh ants, and he saw that also the optimum were not completely aligned for lifespan and egg production.
Good. Okay, I think the rest of the questions we might keep for the panel discussion afterwards. So I will invite Rong now to the screen. Wait. Perfect. All right, Rong. And I will close Audrey's video for now. And
yeah, I can share.
Rong, now the stage is yours.
Okay, thank you.
Perfect.
Yeah, is this okay?
Very good. Thank you.
Okay. Thanks for having me here. Today, I won't specifically talk about foraging behaviors, but I will talk about the behavior state that is immediately following the foraging behavior. So my talk title today is the interface between motivation and consumption in hunger and thirst. Let me change this. Okay. We almost all have the experience in face of high palatable food or beverage. Once we begin to eat or drink, we simply can't resist to eat or drink much more than what we really need. Regarding to it, free will doesn't seem to exist. Consumption behaviors from the sampling to savory cannot be willed by our will. So how does this happen? To understand this question, we need to know first a little bit of background of the motivated behavior. As early as 1854, French physiologist Claude Ballard has already pointed out the importance of maintaining the stable internal environment for all different life forms. Consistent with this idea, maintaining the stable energy, the stable internal osmolarity, the stable temperature, and so on, are therefore the most basic needs for individuals. Behaviors to satisfy these basic needs, such as feeding, drinking, thermoregulation, and so on, are therefore the most fundamental motivated behaviors. They are essential for animals' survival. So understanding how our brain controls this motivated behavior is very important to understand the neuronal mechanism of this motivated behavior, and also provides an ideal entry point to understand how our brain works. According to Craig's framework, all the motivated behavior can be deconstructed into these three pillars. So appetitive phase, consumptive phase, and satiety phase. For simple understanding, I will replace these three phases with three simple words. They are seeking, doing, and done. Seeking phase is namely to seek and obtain the desired outcome. It is the foraging behaviors in the feeding. And the doing phase is usually associated with the engagement of the goal of the behavior. It usually includes the sensory inspection of the desired outcome and also the motor action that is usually associated with the consummating of the goal of the behavior. Usually, the satiety signals is accumulated during this doing phase, which finally let animal enter into satiety phase, that is down phase here. In the case of feeding or drinking, the consumptive phase can be specifically replaced by the eating or drinking behaviors respectively. And these three pillars in the motivated feeding or drinking behaviors can be reshaped to be represented as a closed loop here. Physiological state, such as hunger or thirst, will motivate the animal to enter into this foraging behavior and subsequently eating and drinking behaviors respectively. The following satiety phase will provide the feedback signals to inhibit hunger or thirst sensation. Hunger or thirst state are very unpleasant state, and according to the literature, these states convey the negative valence, which will motivate the animal to enter into this seeking phase. However, hunger or thirst are not the only component that contribute to the motivated feeding or drinking behavior. There also is a feed-forward pathway in which the positive valence that is derived from the hedonic value or the nutrient properties of the food or beverage that are consumed during this consumptive phase can further potentiate the consumptive behavior, which is usually independent of hunger or thirst state. We usually call it hedonic consumption pathway. While a notable study focused on this hunger or thirst circuits in recent years, there is a knowledge gap of understanding the detailed neural mechanism underlying this hedonic consumption pathway. Previous work, including the work from our lab, established that activation of AGIP neurons in mice can greatly mimic hunger state, which can lead to voracious eating behaviors. And the activation of AGIP neurons conveys negative valence. As you can see here, mice will try to avoid to stay in the chamber that is previously paired with the activation of AGIP neurons. And this is the same case in drinking. After I joined the lab, I firstly identified specific activating NOS1 neurons in the subphonical organ can greatly mimic thirst state, which can lead to voracious drinking behavior. And this is also confirmed by another study from the Charles Drucker's lab. And similarly to AGIP neurons, activation of SFO-NOS1 neuron conveys negative valence. Mice will try to avoid to stay in the chamber that is previously paired with the activation of SFO-NOS1 neurons. So according to this, on the hunger or thirst state, metabolic signals or osmolarity signals will activate AGIP or SFO-NOS1 neurons respectively. And this activation will convey negative valence, which will motivate the animal enter into voracious consumption behavior afterwards. So according to this, you will expect it, AGIP or SFO-NOS1 neurons will have higher firing rate during this consumptive phase compared to this no consumptive phase. However, this is not the case in vivo. Both AGIP and SFO-NOS1 neurons tend to decrease their firing rate once mice begin to consume food or water, according to this calcium imaging data. So although activating of AGIP or SFO-NOS1 neuron can lead to voracious consumption behavior, these two types of neurons seems to be only responsible for the consumption initiation, but not consumption maintenance. It will be clear if we look back into these three pillars. On the hunger or thirst state, AGIP or SFO-NOS1 neuron will convey the negative valence, which will work as a teaching signal to motivate the animal into these foraging behaviors and subsequently voracious eating or drinking behaviors, respectively. Some other neural implementations, such as GABAergic neuron in the lateral hypothalamus, will convey the positive valence, which will work as a teaching signal to motivate the animal enter into this seeking phase that is independent of the physiological needs. Satiety signal, such as that conveyed by the PBN CGRP neurons, will be accumulated during this consumptory phase, which will finally terminate this consumptory phase and let the animal enter into this satiety phase. While this seeking phase and the satiety phase have been heavily studied, very little is known how the consumption phase is regulated. Consumption initiation and consumption maintenance are two distinct behavior phases. To understand the neuronal mechanism of the motivated behavior, it's very important to determine the synaptic involvements of different neural implementation underlying these discrete behavior phases. So here, we aim to understand the neural implementation underlying this consumption maintenance. Feeding and drinking behavior are two different motivated behaviors with distinct goals. However, according to Craig's framework, they contain the similar sequential behavior activities across these different behavior phases. So it implies the potential shared pathway underlying this generality. For the fields people study the motivated behavior, people always tend to think of the specificity of different motivated behaviors. Much less attention is paid on this generality. So here, we took an opposite approach. Given the similar aspects of feeding and drinking behaviors, we hypothesize that hunger or thirst circuits will converge at some points to regulate the similar consumptory behavior. Specifically, hunger circuit that starts from HRP and thirst circuits that starts from S1E4 neurons will directly or indirectly through some middle relays to converge at some regions to regulate this similar consumptory behavior. Those shared regions are potentially to be involved in the internal motivation transition, the sensory feedback processing, or even the motor activity coordination during the consumptory phase. So this approach doesn't necessarily map to the regions that is responsible for the consumption maintenance, but at least can greatly help us to narrow down these candidate regions. Now, to make a long story short, through the whole brain search with this approach, we find some shared, direct shared downstream regions, that is directly downstream regions of both HRP and S1E4 neuron, such as PVH or lateral hypothalamus as we expected. We also potentially find some regions that is potentially to be the polysynaptic convergence between hunger and thirst circuits. And among these candidate regions, we notice a hotspot in the hindbrain area. These regions hasn't been fully characterized by any institute reference yet. So here we call it peri-LC because it locates near or even partially overlapped with nucleus, as shown in this crawler section at this position in mice brain. Very little literature has been reported regarding peri-LC, so it's functionally unknown whether this regions is involved in motivated feeding or drinking behaviors. Peri-LC primarily contains vagus neurons, which are intermingled with vagus neurons, and a few neurons with the TH marker in this area, which are the norepinephrine neurons in the nucleus. Chemogenetic silencing of peri-LC vagus neurons can dramatically increase the food intake, and this silencing is rewarding. As you can see here, mice will try to stay in the chamber that is previously paired with the opgenetic silencing of peri-LC vagus neurons. While this project is underway, There are two other studies come out to show that peri-LC might be involved in the salt intake or food intake, indicating a role of peri-LC in the homeostatic control. So comparing to the hunger or thirst state that motivated animal to enter into this seeking phase by conveying negative valence, peri-LC vagus neurons increase appetite with positive valence. So it's more likely peri-LC is involved in this hedonic consumption pathway. Next, we sought to ask which specific behavior phases that peri-LC vagus neurons are involved. We trained the mice to pair this auditory cue delivery of the food or water delivery. So in the friendly movie chambers, upon this auditory cue delivery, the experienced mice will quickly run into the food or water pot to initiate their consumptory behaviors. We focused the in vivo dynamics of peri-LC vagus neurons in these self-motivated conditioning consumption trials. With this head-mounted miniscope microscopy, as you can see here, peri-LC vagus neurons is heavily modulated once mice began to nick. So as you can see here, peri-LC vagus neurons are bidirectionally modulated during the consumptory phase. They are both activated and inhibited response once mice begin to consume food and water. And this response is better aligned with the first nick, but not aligned with this auditory cue and the delivery of food or water. I don't have time to show the details here, but our imaging experiment demonstrated that peri-LC vagus neurons are specifically involved in this consumptory phase. And with the inhibitor response, these neurons carry the scalable pandability signals, which is determined by the hedonic value of the consumed food or beverage. And this signal can be modulated by the metabolic states such as hunger or thirst, which can induce the pandability shift in face of the same food or beverage. The scalable pandability signals carried by the peri-LC vagus neurons can also be behaviorally reflected by the consumption bout durations. So according to this, we can further hypothesize that inhibiting peri-LC vagus neurons just within the consumption window will lead to the behavior consequences that are associated with the pandability shift. In light of this, we designed a new perturbation behavior paradigm in which mice is freely moving in this behavior chambers and under self-initiated consumption trials. Diluted insure, which is modest sweet, is given throughout the whole session. With the contingent behavior paradigm, the short pulse photoinhibition of peri-LC vagus neurons is triggered by the nick events. With the same animal, we also include this non-contingent behavior paradigm, in which the number and duration of the photoinhibition is yoked to the contingent behavior paradigm, but it's totally independent of the nick events. So with this randomized short pulse photoinhibition, we sought to ask whether inhibiting peri-LC vagus neurons outside of these consumption windows will change this consumptory behavior. We also include this sham behavior paradigm, which is blocked as a control here. As we expected, only under this contingent behavior paradigm, it can dramatically increase the food intake, which contrasts for the non-contingent and sham behavior paradigm. There's not much difference between this control and the perturbation block. Next, we focused on the consumption bout durations that mice is spending in the food zone area. Interestingly, while the consumption bout numbers haven't been changed across this contingent, non-contingent, and sham behavior paradigm, the consumption bout durations is dramatically increased only under this contingent behavior paradigm. And this is consistent with our observation that peri-LCA growth neurons are only involved in the consumptive phase, but not appetitive phase. So the activity of these neurons are only responsible for the consumption maintenance, but not consumption initiation. And inhibiting of these neurons can prolong this consumption duration, but won't change the frequency that mice approach food. So in our story, during this consumptive phase, consumption of food or beverage will inhibit peri-LCA vagus neurons. And with this inhibitor response, these neurons will carry these pathogenic signals, which will feed back onto the ongoing consumptive behavior and therefore prolong this consumptive duration. So these complementary properties of peri-LCA vagus neurons and the consumptive behaviors is indicated to be a double negative feedback relationship, which is functionally similar to the positive feedback. Physiological states such as hunger or thirst can further modulate this double negative feedback, which can induce this pathogenic shift in face of the same food or beverage. A classic view to look at the 3 pillars in the motivated feeding or drinking behavior somehow resemble these 3 states in the Markov chain model. In this model, in every moment, there is a probability to stay on the current state and also probability to transit to the nearby state. However, unlike the Markov chain model, in the ideal situations, the 3 pillars in the motivated feeding or drinking behaviors is unidirectional. So in every moment, there is a probability to stay on the current state and also the probability to transit to the following state. Our experiments indicated that peri-LCA vagus neurons with the inhibitor response encode the probability to stay on the consumptive state. Now, to our knowledge, this is the very first time to show the neural implementation to encode a specific variable during this consumptive phase. So under contingent inhibition of peri-LCA vagus neurons, this p-value is changed. So within the consumption part, as time lapses, the probability to stay on the consumptive state is dramatically larger compared to the control, and therefore prolong the consumption duration. Now, in summary, we firstly identified a novel regions, peri-LCA, as a polysynaptic convergence of hunger and thirst circuits. Peri-LCA vagus neurons conveys the pathogenic signals with the inhibitor response. And with this inhibitor response, these neurons will maintain the food and water consumptive phase through double negative feedback. And what's more important, peri-LCA vagus neuron is indicated to be involved in the hedonic consumption. The characteristics of peri-LCA vagus neurons on this palatability modulation and enhancing food or water consumption provide a new insight into this hedonic-driven consumption. It is potentially represented as the neuronal underpinnings of overconsumption in face of high palatable food and beverage. As I mentioned in the very beginning of this talk, consumption behaviors from the sampling to savory cannot be viewed by our view. There are specific neural implementations underlying this phenomenon. The property of peri-LCA vagus neurons on this pathogenic-guided consumption validated as an ideal entry point to study this hedonic-driven consumption. Now, back to the 3 pillars in the motivated feeding or drinking behaviors. With our study, this picture can be more intact. So under hunger or thirst state, the accumulated signals that are carried by the AGRP or S1E4 neurons will motivate the animal to enter into this foraging behavior. And this maintained activity of AGRP or S1E4 neurons will maintain animal on this seeking phase. The availability of the food or water in the environment during this seeking phase will trigger a motor command from the motor cortex or basal ganglia to initiate this consumptory phase. And this consumptory phase is maintained by the activity of peri-LC vagus neurons. Satiety signals, such as that carried by the PP and CGRP neurons, will be accumulated during this consumptory phase and work against with peri-LC vagus neurons, which finally terminate this consumptory phase and let animal enter into this satiety phase. However, these unidirectional three pillars in the motivated feeding or drinking behaviors only exist in the ideal situations. In the naturalistic feeding or drinking behaviors, the situation is much more complex. The behavior states can often be reversed across different behavior phases. For example, motivation competition from other internal needs can be motivated animal to terminate this foraging behavior and enter into this satiety phase. Sensory feedback, such as the unpleasant taste during this consumptory phase, will motivate the animal to terminate this consumptory phase and drive back into this seeking phase to further search for proper food or beverage in the environment. And even within this consumptory phase, it also can be potentially intermingled with the short period of no consumptory phase that is often associated with the coordination with other motor activities. Such as that cleaning up mouth with the grooming behavior during the consumptory phase, or catching up the breath during the consumptory phase. So it would be of significant interest next to know how these different behavior phases is initiated and maintained under such a complex environment in order to guarantee our correct execution of motivated feeding or drinking behaviors. The dysfunction of any neural implementation underlying these three pillars will greatly change our consumption behavior. So understanding the selective involvement of these neural implementations across different behavior phases not only help us to understand the neuronal mechanism of this motivated behavior, but also potentially provide a new insight into the mechanism of the different disorders. In the case of feeding, it could be different kinds of metabolic and eating disorders. So with that, I want to thank all the lab members from Scott Stenson's lab, especially my mentor, Scott Stenson, my collaborator, Senjin Xu, and Hamstand from Genelia, and Yang Yu from Allen Institute. I also want to thank the people from the Genelia facilities who have ever been involved in this project. With that, I'm open for the questions. Thank you.
Thank you so much for your fascinating talk. I actually wanted to ask a quick question in the beginning. So you were talking about the sensory input during the consummatory phase, being able to kind of reverse the consumption back to the seeking pillar.
Yes.
Is there anything known about like potential feedback connections between the peri-LC neurons back to the AGRP neurons, for example?
I don't think there's a potential connection in that case. Actually, even this, for example, nutrient property or hedonic value, which can drive back into this seeking phase, that is totally independent of the activity of AGRP or S1E4 neurons. So it potentially is through other neural implementations. We don't have the solid data to show which neurons or which circuits they are, but presumably will be like, for example, like GABAergic neurons in the natural hypothalamus, or the VTA stuff, dopamine neurons, such like that. But for sure, it's totally independent of hunger or thirst state.
I see.
Yeah.
We have a question by Laura. In the model, it's assumed that the mice consume to satiety. Is that always the case? If there are alternative options or other pressures, such as other mice or predators, does this change? And do peri-LC neurons change their activity?
Yeah, I think I mentioned a little bit here. Whether you can see my screen? Do you can see? Yeah, okay. So yeah, this is a ideal situation, right? This is a unidirectional pathway, right? That's the ideal situation. But in the naturalistic feeding or drinking behaviors, there also always a drive back to previous behavior state. For example, the motivation competition from other internalists, such as the predator approach, which Nora just mentioned, right? We are terminated this animal to we are terminate this seeking phase and drive animal enter into this satiety phase again, because they need to quite to think about the specific situations at the moment. It's better to escape instead of still try to hunting something for it, right?
That's true, yes.
Yeah.
Okay, we have a question by Shambhavi Shidambaram. Sugar has an effect on satiety in humans and reduces it, leading to an increase in consumption. Could that effect happen via the peri-LC VGLUT2 neurons?
Are you asking, sorry, I cannot see the panel, but I'm supposing you are asking whether it can increase the overconsumption if we perturb, manipulate the peri-LC VGLUT2 neurons activity. Is this the question?
Let me go back to it. So basically, the question asks whether sugar having an effect on satiety in humans could happen through the peri-LC VGLUT neuronal circuitry.
Oh, definitely. Yeah, sorry, I lost your question, but definitely, because we talk about the palatability information. So here, I don't have time to talk about the details here, but our experiment indicated that this palatability information is derived from the hedonic value of the consumed food or beverage. So I think sugar, because sugar is sweet, definitely it has a very high hedonic value for the animals. So I believe it definitely can work on this peri-LC VGLUT2 neurons pathway.
Great. Okay, I will invite back Audrey to the screen.
Okay.
As well as...
could you help me to stop share? Because seems
I think you should stop sharing.
Oh, okay. Okay, great.
Yes. And let me also invite Charlotte to the screen. Okay, so I think that is all of us. Great. Okay, so now the idea is that we start a bit of a more general discussion in terms of how internal states shape foraging behavior or feeding behaviors in general. We have specifically a guest panelist today, Charlotte Rocher. Maybe you want to start off, since I can maybe also go a little bit through the questions later.
Sure,
but if you have any specific questions to the speakers, you can start.
Yeah, thank you both so much for great talks. I was kind of thinking about the intersection of both of your talks, actually. I wanted to ask Ron Gong, how do you think the peri-LC neurons and this kind of increase in that consumptive phase also interacts with like deficiencies and things like we also saw with the answer, which completely different context, of course, but how do you think the sort of that increase in eating, literally just keep eating or drinking, will also have inputs from maybe when there's an excess of something or a deficiency of something?
Yeah, I don't. We don't look that far so far. So
yeah,
the direct link through the peri-LC to the overconsumption, this link we didn't establish that. We just say this peri-LC can prolong this consumptor phase. Of course, overall will increase this consumption. But whether it's this dysfunction or abnormal activity of the peri-LC vagus neurons can indeed increase the overconsumption, which finally leads to the obesity. That link, we haven't established that, but we hopefully it will come in that way, and we are still studying these variants. So, did this answer your question? So I'm.
Yeah, absolutely. No, I thought it was really interesting in the ants, the way actually, although they have all these social features, actually they were quite often out to get the best nutritional things for themselves as well. It's fascinating.
Yeah, I guess it's very interesting, like, because Andrew talked about the internal states will determine which specific nutrient you want to search for, right? This is come from your internal metabolic state. That is, give the internal motivation, the internal drive to search for specific nutrients. But when you finish your foraging behavior, for example, you have already found this food, and you need some specific, like a quick sensory feedback to keep you stay on this consumptive state until you satisfy your physiological needs, right? So that's my study focus on. So kind of like a bridge the study with just following the behavior physics that Andrew is trying to study, right?
I have a kind of a follow-up question on this. So
yeah,
I was thinking, kind of looking at both talks, whether it is, I mean, it's interesting to see, right, this happening in two very different species and like evolutionary distant species. And I was wondering if maybe in the nature of foraging, that leads to basically a similar logic for some of the controls. So what I mean is that you have certain decision sort of sequence or hierarchy, if you want. So for example, the separation of a decision to seek, right? Like while you're foraging, like walking around, whereas then decisions on the food that are about whether I keep consuming or leaving, and at which point, for example, the state comes in and like which parts of this longer sequence are modulated by internal state, and whether maybe because once you're on, like you can only fully evaluate the food source when you're on the food, at which point you've started consuming. So it makes logical sense to have the state come in there. I don't know if you can comment on this.
So the ants, I think they go out. So you can see when you create a deficiency, all the extra needle workers are outside because they lack something. So they're very motivated to forage. And at the beginning, they're completely blind. So they just go because they're very highly motivated to find something that is missing. And when they arrive at the food source, the behavior is quite like specific. They would touch the food source. And as soon as they touch, if they like it, they stay on the food. And the duration of a feeding bout doesn't change as a function of the quality of the food. It's just a decision, okay, I feed or not. And if they don't like it, they stop straight away and they go out. So I think they go out blindly. If they found a good food source, that's good. They stay there, they're in foster trade. If they don't find a good food source, they go out and they try to find something else. So at the beginning, they're just motivated to find something, but they don't know about the food source at all. And they don't compare the food source anyway. Most of the ants, they would perhaps visit the food only once during the experiment. So it's really a collective, the choice emerged at the collective level. But the decision of reinforcing the trail is an individual, it's at the individual level, and which is the one that is in fact playing on the collective level.
So a quick follow-up then on this, maybe for sort of the ecology, is that they generally, do they eat from the same food source for a long time, or are they relying on very sort of volatile food sources?
So for the Nepithema or black garden ants that I study the most, they have the same food source every day. So they would collect honeydew from aphids, and honeydew is rich in amino acids and sugar. And so the aphids are always at the same spot. It's a permanent food source. Linepithema humidae is more like an opportunist. It will find prey somewhere, it would feed on aphids. But Lasus niger, it's really a species that would go on permanent food source. So, but they experience a lot of different food sources in the field. So both of the ants study are generalists, so they would feed on a lot of different food sources. So some are what we call ephemeral, it's like a prey, but the aphids are considered as permanent food source. Yeah.
But they always have to go to both food sources in the assay to be able to.
No. In
Sorry,
fact,
it was more about the first question.
No. Some ants they just visit the food source perhaps 7 times during the assay. Some just once. In fact, the ants going out and always follow the trail, and so it pick when it arrives at the fork. It picks the highest concentration for the trail. You know, they have these 2 concentration in pheromones, and they pick the one with the higher concentration. They follow. They arrive at the food source. They like it. They stay there. They don't like it. They go back, and they go back to the nest or to the other food source. This is completely random too. So it's really the fact that there's a lot of ants playing that you see this collective choice emerging. But ants do not have to compare this, the 2 food sources to make, in fact, the appropriate decision, which is why it's very interesting, in fact. It's emerging from the collective.
Is there anything known about information transfer within the nest or colony?
Yeah, in fact, when an ant is coming back, he has to empty his social stomach in another ant, the atrophylaxis. And if the other ant doesn't like the food source, he won't take it. And an ant won't go out if she can't empty her stomach. So if she can't empty her stomach, if nobody wants the food, she would stay in the nest and she would stop foraging, and another ant would go. So there's also a transfer information like that, because when they transfer the food, they can estimate also the one inside the nest, the quality of the food source that's coming back. And right now, there's a team. The team of Pheromone develop a very interesting techniques where they can, with fluorescent markers, really track the food, not the ants this time, but the food, the liquid inside the nest, and how it's mixing between ants and which ant is asking for what. So I think it will bring a lot of insight in the next future about how, because we don't know exactly how it works inside. So because we just see 10% of the colony all the time outside. So
yeah, it's difficult to measure, I guess. We also have one question from Laura. Are there any longer term, such as seasonal changes on how ants balance their nutritional needs?
Yeah, Spence Beamer and Steve Cook did this experiment on fire ants, and they show that the intake target change according to seasons, because ants have to prepare for hibernation, and so they don't forage for the same food source for winter. And also because the egg production rate changed across the year. So when they are producing a lot of larvae, they collect a lot of protein. And so, of course, yeah, it changed across seasons.
I was going to actually ask a similar question for Rong as well, because the time scale of the neurons that you're recording, are they always going to only modulate behavior on the basis of one consumptive bout, for example, or if there are seasonal changes, mice in different environments or different habitats over longer periods of time? I know you don't have data yourself on this, but do you know about anything that might be sort of relevant to this?
Yeah, I can speculate a little bit about it, because the seasonal change, oh my God, in mice. I never think about that. Yeah, I only say the inhibitor response during the different consumption part. Actually, the positive correlation between the consumption part duration versus the inhibitor response amplitude of the periodic vagus neurons is very obvious. So basically, you can, according to the neurons activity of the periodic vagus neurons, to predict the consumption part durations. Use neural activity to predict the behavior. And also, I believe vice versa, even though we haven't done that, decoding, encoding, but it's very clear. If you are talking about long-term changes, so I guess there must be some plasticity happens there, because we did long-term inhibiting this neurons activity. For example, 1 month or 2 months inhibit the neurotransmitter release from these groups of neurons. We actually didn't observe any effect. So this is quite contradictory from our short pulse inhibition results, right? So, and many people ask about this. How do you explain this contradictory part? Well, actually, I think there's quite common for the brain system, because there's definitely some redundant pathway happens there, right? There's some plasticity. Even if one part of the brain is silencing for long time, like 1 month or 2 months, there must be some other parallel pathway pops up, or even have previously have the weighted collection, right now have much stronger weights in this behavior, so it can compensate for these deficits. Long-term changes, I'm thinking about, I also think about for the obesity populations. For the mouse, for the laboratory mouse, for example, the background such as C57, it's very hard to select out the natural fat mice from the laboratory breeding, right? But if in other species or even other strains of the mice, it's possible to select out 1 or 2 like really obese mice. That is not, I'm not here talking about the OB mice, but just the naturally obese mice. And if we can check whether these mice, these peri-LC regions has some abnormal activities compared to the other mice, and also correlated with their obese phenotype, maybe that's really interesting part for the long term. Yeah.
I have a related question.
Yes.
So it's about essentially, okay, I can basically imagine 2 sort of strategies to basically maintain homeostasis and fill like your current metabolic needs. One is sort of, I'm just going out randomly and checking each food source I encounter if they are good and then consume or not. And the other one would be to build on prior knowledge I built up to actually have a more directed seeking, right? And so that relates to also what do we know about plasticity related to like basically learning which food sources have a specific content, right? I assume that's mostly learned, or especially maybe like longer distance cues for navigating there would be learned. And it seems like that probably both processes happen at the same time, but I don't know what you know about how much these matter for the species you study. So was this clear?
Yeah, it's very clear. I just want to, I don't have specific answer for that, but I want to comment a little bit about it. Because we always talk about consumptive behavior, including the motivated feeding, drinking behaviors, of course, including these foraging behaviors, consumptive phase, and the satiety phase. All of these behaviors are actually the learned experience. Definitely, it's had a hardwired component, but also has the learning component to ensure this correct execution, right? And this behavior is greatly shaped not only the internal states, but also the external information. External information will come from the like available of the food or water in the environment, and the internal states that's Andrew studied, like you are deficient for specific macronutrient or micronutrient. And all of this working together to greatly shape our consumptive behaviors. For the internal state, some of this internal state arises due to the physiological changes. So, for example, I will talk about hunger or thirst many times in this talk, but there are also other physiological changes, such as like arousal state, emotion state. All of this can modulate their foraging behaviors, their consumptive behaviors. And besides that internal state, there's another type of internal state that is the consequences of the past experience, which people usually call it internal references. That is what you just mentioned, like learning plasticity, right? Experience from the past will spontaneously shape your current behavior and even determine your motor actions in the future. So I think this experience greatly determined our food or beverage preference, especially in human, right? Like think about like why do you specifically recall which food you like most and which food you specifically like drive to a specific restaurant and specifically call some specific dishes and eat a lot of it even beyond your physiological need. So yeah, I just think all of this information should come together, no matter in the foraging behavior phases or the consumptive behavior phases.
We have another question by Chris Rogers. How do the foragers sense the needs of larvae? Is it directly communicated between individuals, or do the foragers respond to whether the food they bring is accepted or rejected? So maybe more on a developmental scale, how are things communicated?
So there's like a different hypothesis about larvae, but to be honest, nobody knows really well. Some people are saying that there was a kind of a brood pheromone that is expressed by larvae when they are very hungry. So, and that ants can sense that. Some people say that they are moving, they're quite excited like that, and they're moving a lot. And so ants know that they need to feed their larvae. And larvae can accept or refuse some food source. So you know that ants can learn, okay, I brought that and they didn't say yes, so I'm going to bring something else. So of course, ants also can learn, coming back to the learning, of course, ants can learn what was like, if they were successful in a foraging trip or not successful in a foraging trip. So anyway, larvae usually communicate their hunger state to the nurses, and nurses communicate this hunger state to the forager. So it's a chain of demands inside the nest. So we don't know exactly how, like which methods they use, and I guess it depends on the species. So for instance, in green-headed ants, it was very intriguing because the larvae were acting like a social stomach. So meaning that ants were not feeding on the food at all. They were bringing these bunch of food, these crumbs, and they would give it straight away to the larvae without feeding on them. Larvae would feed on it and regurgitate foods directly to the adults. So in fact, adults were using the enzyme produced by the larvae to digest the food, so they didn't have to do it by themselves. So it was really that kind of a division of labor of digestion too, which was quite intriguing. So larvae plays a big role within the nest.
Wow. Is there any information that is kind of communicated within different life cycles of the colony in the wild? Is there anything known about? So the colony, I mean, basically, the ants of a colony do not live forever, right? There is a life cycle of the colony going on. Is there any information about nutritional states of this environment transferred from one life cycle to the next, for example?
Yeah, so a colony of ants can live up to 10 years, for instance, in Dussutour, but an ant is true, live only for 2 months in the field because only the queen lives for the 10 years. And so, of course, workers are produced all the time, and workers go through what we call age polyism. So they started being nurses, and after they become a forager when they get old. So, of course, they get experience during their life cycle. At the beginning, they stay inside the nest and they receive food from the other, and after they are the one getting out and collecting the food. So, of course, even their nutritional needs as workers changing across the life. Because we measure the fat content, for instance, between nurses and foragers. Foragers are very lean individuals, super skinny, while very young ants are nurses and they're super fat. They have like 25% of fat, and foragers have only 5%. And if you manipulate fat content, you can extend lifespan of foragers, for instance. So if you take an old forager, you put it on a fat diet, you will extend its lifespan like that. So having, at least in ants, being obese is quite good.
That's great to hear.
Kind of following that kind of thing, if you leave your experiment running for over a longer time, and you had the intranodal ants, when they're not being given the food that's actually suitable for them, they will leave. Do they come back and resume their roles when they are sufficiently fed?
Yeah, usually yes. Yeah. We check that. Usually they come back to the nest, and they really get out only when they're very unhappy. But we've seen in this experiment, when you starve the colony of multiple nutrients, even the queen sometimes will get out. if she doesn't find what she
Okay.
needs. So just in Argentinians, because there's the queens are very mobile, but we've seen them in experiments because there are multiple queens in an Argentinian nest. And sometimes we've seen the queens getting out and say, oh my gosh, she must not be very happy, must be very hungry.
Those workers aren't doing their jobs.
Exactly. But yeah, so ants, when they're like, usually intraneedle workers, they have a, it's a question of threshold. They don't react quickly. Except if you reach a threshold, like, okay, now I'm too hungry and nobody's bringing the food I want, so I'm gonna get out my myself.
It's interesting that, like, despite sociality, I wonder if, like, the circuits that are really, like, not social and are all about yourself kind of can override it.
Yeah, I mean, the social stuff is the fact that you bring back the food to the oven. But it's funny to see that because the question was, even do the foragers listen to themselves or to the individual? And we were very surprised to see that they listen first to themselves. I'm first taking the food that is good for me, and I will see after for the other one. But I thought that at the beginning with Steve, we had the idea that perhaps the foragers would go for the food that the workers inside the nest wanted. But no, first they thought about themselves. So yeah, it was a bit like selfish behavior at the beginning.
All right, so we slowly approached we already approached the 1.5 hour mark. Maybe I end up with a comment from our first speaker, Alex. The negative feedback models are homeostatic and consider deficits as aversive. I jumped. In fact, the act of decreasing a deficit can be hedonically positive, hence making deficits appetitive. Animals like humans may want to want. An obvious example is libido, which humans value because it creates a need, even if it doesn't reflect any deficit. The motivational system evolves to invent itself needs that are evolutionarily beneficial. That's a comment by him in terms of the hedonic values.
Yeah, I think to some degree, I agree with this comment, because our model doesn't definitely exclude the positive feedback, right? When we talk about like hedonic value or nutrient properties of the food or beverage, it can drive back to the seeking phase. That is actually, we call it hedonic consumption pathway. I think it's similarly to what you call like want to want, this positive valence. And correct me if I'm wrong, but I think there's a similar sense.
Okay. I think with that we can close our session. We basically, the remaining questions we could also answer offline then. So thank you very much, everyone, for joining us for this episode. Thank you, Audrey and Rong, and as well as Charlotte for joining the panel discussion. And I hope to see you all in 3 weeks for our 5th episode then. Thank you. Bye-bye.
Thank you very much.
Yeah, thank you.
Bye.
Thank you.
Bye.
Bye.